Now, having said that around 2/3 of most things are reabsorbed, there are some things which are pretty much 100% reabsorbed by the end of the proximal tubule, and I'm thinking in particular of glucose and amino acids. Now, obviously, glucose and amino acids are things that the body wants to retain. We work very hard to obtain those in the body, tracking them down, eating them, absorbing them. We want them, and we definitely don't want to lose them in the urine. So the proximal convoluted tubule expends some energy in order to make sure that they're completely reabsorbed and under normal circumstances don't get through into the urine and are not lost by the body. Now, the problem is, if you want to completely reabsorb something, ultimately, when it's nearly completely reabsorbed, you're going to be absorbing it against a concentration gradient. Now, that's theoretically a problem, because if you want to move glucose against its concentration gradient, from a theoretical point of view, you have to put in energy. The way this works is through secondary active transport. And the way secondary active transport works is that we would have a cell, in this case it would be a proximal convoluted tubule cell, and on its luminal membrane, which is the membrane which faces the tubule lumen, we would have an SGLT transporter. Well, lots of them really. The SGLT transporters are sodium-coupled glucose transporters. The idea is that they transport sodium into the cell and they transport glucose at the same time, so they are sodium-glucose symporters. There are different types of SGLT transporter. Some of them transport 2 sodium ions for every glucose, some of them transport just 1. But I won't distinguish between those here. The essential concept is that if sodium is allowed into the cell, that's moving in down its electrochemical gradient, and that releases free energy. Now, the transporter has a way of coupling that energy release to the uptake of glucose. So in effect, sodium is moving downhill and glucose is moving uphill. And the movement of sodium downhill helps pull in the glucose as well. Now, this uses energy. The energy is in the electrochemical gradient for the sodium, and ultimately that gradient has to be maintained, and that's maintained using a sodium-potassium ATPase, the sodium pump, which is found on the basolateral membrane of these cells. The basolateral membrane being the other membrane that isn't facing the lumen, that is forming the sides and the end of these particular cells. Now, the sodium pump uses ATP. So this is a primary active transporter. It's expending ATP energy in order to maintain that sodium gradient, but as a result, sodium wants to come into the cell. If it's allowed in, it releases the free energy, and that's used to energize the uptake of glucose. So ultimately, it's an energy-consuming process, but because the SGLT transporter is not using ATP directly, it's referred to as a secondary active transporter. And it works very effectively to reabsorb under normal circumstances all of the glucose from the filtrate, so that essentially none of it remains in the urine.